Within the eukaryotic chromosome, the DNA is held together by histone proteins. So together, the DNA and histone proteins are called chromatin. Now, when the cell is at rest, or not really at rest, but when it's not actively dividing, DNA takes this form, chromatin. It's loosely coiled. It's not packed tightly into what we call a chromosome. So once that DNA is wound around the core of 8 histone molecules, we call that a nucleosome. And nucleosomes join together and form what we call euchromatin. U meaning true. Euchromatin represents active chromatin that can be transcribed by RNA polymerase and transcription factors. I know that doesn't mean a lot to you right now because we haven't really learned much about the process of protein synthesis, which includes transcription. But we'll just say like this: euchromatin can be used to, can be translated basically into the code from the DNA can be translated into an actual protein, because that's what DNA does. DNA codes for specific proteins, and that is how it leads to DNA containing the genetic material that gives us our traits that we have, our genetic traits. It's literally by DNA coding for individual proteins. So if the chromatin is in a form called euchromatin and it's not tightly packed, then it's active and it can code, it can work to synthesize a protein. Okay, but, and this is just an image of it. So this is naked DNA, the DNA double helix, wound around histone proteins. And each one of these pieces of DNA wound around 8 histones is called a nucleosome. And all of these nucleosomes pack together in a loosely coiled form called euchromatin. But when it packs together tightly, we call it heterochromatin. And heterochromatin cannot be translated into a protein. It cannot be used to produce a protein or synthesize a protein. It's inactive. And then heterochromatin packs even more tightly to coil and form into chromosomes, which is what we see during the M stage, during mitosis. Okay, and here are the notes on that. A more highly compacted form of the chromosome is heterochromatin, which is inactive chromatin, and the genes in heterochromatin are hardly ever transcribed because of the fact that the DNA and the histone proteins are packed so tightly together. This is a table on slide 17 that lists some organisms, some common organisms, their scientific name and their common name, and the number of chromosomes in one cell of that organism. So we've got Saccharomyces cerevisiae, or yeast, like that's used to make bread and beer, has 32 chromosomes per cell. You go down here to humans, or Homo sapiens, we have 46 chromosomes per cell. Very interesting, the southern adder's tongue fern, Ophioglossum vulgatum, has 1,320 chromosomes per cell. The one that has the closest number to humans off of this list anyway is the potato. Solanum tuberosum has 48 chromosomes per cell. So the number of chromosomes per cell is not necessarily an indication of how closely related the organism is. But this just lets you know that chromosome number is different in different species. So with chromosome number, no matter what species or what organism that we're looking at, the diploid number, or the 2N number, is the number that includes 2 sets of chromosomes of each type. Humans have 23 different types of chromosomes, but each type of chromosome is represented twice in each body cell. That's the diploid number. What that means is that we get 23 chromosomes from one parent and 23 from the other parent, and that gives us the 46. Okay, so our diploid number in humans is 46. The only cells in humans that have the haploid number, which would be 23, are sperm and eggs. So the haploid or N number for humans is 23, and the diploid number for humans is 46. And again, one set of the 23 chromosomes comes from the father, and one set comes from the mother, the father's sperm and the mother's egg. And that's what gives each of our cells the diploid number, which is 46. Now, chromosomes are duplicated. They go from consisting of a single chromatid to consisting of two sister chromatids at the end of the S stage during interphase. During the S stage, each chromosome is duplicated and consists of two identical double helix DNA molecules, or they're called sister chromatids, two strands of genetically identical chromosomes. They're attached together at a centromere, like we went over in the last recording. And this shows you what a duplicated chromosome looks like again. It has two sister chromatids attached at a point called a centromere. But what we also have labeled and shown in this illustration are the kinetochores, which form on either side of the centromere. These are points of attachment because during the stage that we'll learn as prometaphase, spindle fibers will attach to the kinetochores, and they're going to be used to move the chromosomes around, to line them up, and then to separate them during mitosis. All right, so the centrosome is the centrosome. This is a new structure that we're talking about, is just outside the nucleus, and the centrosome organizes the microtubules or spindle fibers. Okay, so in animal cells, the centrosome contains two barrel-shaped centrioles that are basically lined up perpendicular to each other, at right angles to each other. And the centrosome in animal cells contains centrioles, but in plant cells it does not. But what the centrosome does is organize the mitotic spindle. And the mitotic spindle is going to line up the chromosomes and then help them to separate so that each daughter cell gets a copy. All right, these are the stages of mitosis, or the phases of mitosis. We'll begin with prophase. And one piece of advice when you are learning the events that happen during each phase of mitosis, prophase is the most important one to learn because prophase has the largest number of events that you have to learn that happen, okay? So you've got like 5 things that are going on during prophase, and you need to know all of those events, all of those things that happen, you need to learn them. But if you'll learn them, the other phases are very easy. They either have just one event, or when you get to telophase, you can just reverse everything that you learned about prophase. So spend the most time on prophase and learn the events that occur during prophase. One thing is that the chromatin condenses. Chromatin condenses and forms into chromosomes. So under a microscope during prophase, you can see the chromosomes. You would be able to, in a human cell, distinguish those 46 chromosomes. And those 46 chromosomes would be composed of 2 sister chromatids held together at the centromere. So they would be duplicated chromosomes during prophase. Another thing that happens is inside the nucleus, the nucleolus disappears. And it's not a magic show. It disappears, but it's because the components of the nucleolus dissolve in the fluid inside the cell. Another thing that disappears or dissolves is the nuclear envelope. The spindle begins to assemble. That means we start to see spindle fibers forming, extending away from the centrosomes. And the centrosomes are going to start to move away from each other. They're going to make their way to opposite ends of the cell, because that's where they're going to be when they carry out their role of cell division, of mitosis. So again, chromatin condenses, nucleolus disappears, nuclear envelope disappears, the spindle begins to form, and the centrosomes move toward opposite poles of the cell. Learn those events, because the rest of these phases are simple. Prometaphase, the spindle fibers attach to the kinetochores. Let's go back. The fibers attach to these kinetochores. For every one of those chromosomes, those duplicated chromosomes, a spindle fiber is going to attach. Then we have metaphase, which is when the chromosomes are pulled around by those kinetochore fibers or spindle fibers, and they are forced to align across the metaphase plate or the equatorial plane of the cell. In other words, they line up at the middle of the cell. So during metaphase, chromosomes they form a single file line at the middle of the cell. And then during anaphase, the sister chromatids separate and they're pulled toward opposite poles of the cell by those kinetochore fibers or spindle fibers. So we have two phases listed on this slide, metaphase and anaphase. Metaphase, the chromosomes line up in the middle. Anaphase, the sister chromatids separate. Then we have telophase, which if you learned the events that happen in prophase, telophase is easy because it's just the reverse. In prophase, the spindle appears. So in telophase, it disappears. In prophase, nuclear envelopes fragment or disappear. Now they reform around the daughter chromosome sets. And then there are now two clusters of daughter chromosomes rather than one single cluster of chromosomes. Now there are two sets of chromosomes basically, because there are going to be two cells, and each cell needs to have a set of its own chromosomes. Nuclear envelopes reform. Remember, in prophase they fragment. Now they're going to reform, which we said that up here. So the nucleolus will reappear, which I don't see listed here, but the nucleolus will reappear inside the nucleus. Cytoplasm will divide, and that's called cytokinesis. That will happen alongside of telophase. It happens simultaneously. And then the chromosomes that had condensed, the chromatin had condensed and become chromosomes, now is going to become diffuse and form back into chromatin. So opposite of everything that we saw during prophase. Here's a picture of the top shows microscope images of mitosis happening in animal cells. And then the bottom set is microscope images of mitosis happening in plant cells. What you see happening with the chromosomes is the same. The only difference is cytokinesis is different in animal cells and plant cells. So let's look at early prophase and then prophase. We're going to start to see, mainly what we're seeing is the chromatin, which had been very spread out and diffused. You can't make out chromosomes here. Or here, you're going to see it start to condense. The chromosomes will become thicker, more condensed. You can start to recognize them as rod-shaped structures. They look like kind of like spaghetti strands, I guess, but you can identify them. You can see them. And that's going to happen by the end of prophase. You're going to see these darkly stained chromosomes that are very clearly visible. You also start to see the nuclear envelope. You can really see it here in this plant cell. The nuclear envelope is going to start to dissolve as well, and so is the nucleolus. And here, this must be the end of prophase because I can see the centrosomes. This would be where a centrosome is located, and here's another centrosome. And they're